Good afternoon. I'm Xinpeng Zhang from the University of Nebraska-Lincoln. Thank you for the opportunity. Today, I will talk about our biochemical tools for CAZyme genome mining. CAZymes are enzymes for carbohydrate metabolism. We know that plant and algae can use photosynthesis to turn carbon dioxide into sugars. Sugars can be polymerized into storage polysaccharides, like starch. In fact, most carbons fixed by non-crop plants and algae are turned into structured polysaccharides, like cellulose and hemicellulose, which are the main components of plant cell walls. Glycotransferases, or GTs, one class of CAZymes responsible to polymerize sugars. Now, in biofuel production, cell walls are often referred as lignocellulose and the feedstocks, while in food science, the polysaccharides in cell walls are known as dietary fibers. Depolymerization of these cell wall polysaccharides will need glycosidic hydrolysis, or GHs. After that, we get simple sugars, which can be converted into beneficial metabolites, such as short-chain fatty acids in human gut, or into bioethanols, which are the most well-known biofuels that are mixed in gasoline. Because of this, CAZymes are extremely important for global carbon recycling, dietary fiber utilization, human gut health, lignocellulose degradation, soil health, and plant disease. Now, with so much DNA sequencing data from microbiomes or plants, we wanted to address the question, how do we computationally identify the CAZyme sequences? For that, our lab have developed dbCAN as a computer software system that can take genome sequences as input and produce the CAZyme sequences as the output. dbCAN was first published in 2012. With the NSF grant, we continued to the development of new computer tools centered on dbCAN, which aims to develop new tools with NIH grant to facilitate the use of personalized dietary fiber for improved human health. If you think about what you eat every day, there are a high diversity of dietary carbohydrates in our foods, from milk to veggie to fruit and meat. These carbohydrates exist as various polysaccharides and glycoproteins containing a high diversity of monosaccharides and of their linkages that connect them into glycan polysaccharides. So, what exactly do CAZymes do? CAZymes target glycosidic linkages. These sugar-connecting linkages are called glycosidic bonds. Breaking those bonds need carbohydrate-active enzymes, or CAZymes. Each of these different glycosidic bonds will need a different CAZyme. Therefore, to fully break down complex carbons into simple sugars, countless CAZymes will be needed. Shown on the left is an example of GH. It takes the polysaccharide as substrate and cleaves one sugar out of it. The degree of polymerization is reduced by 1. The glycosidic linkage is basically the carbon-oxygen-carbon bond formed between two sugars. On the right shows 3 disaccharides. They all have glucose, but connecting with different sugar with different linkages, we will get very different disaccharides, for example, maltose, lactose, or sucrose. In our everyday diet, there is a very high diversity of plant-derived polysaccharides and their glycosidic linkages. Here, different colors and shapes represent different sugars. Polymerizing them with different linkages will need different GTs, and breaking them will need different GHs. The CAZy database collects all the experimentally characterized CAZymes. They regularly curate biochemical data and collect their protein homologs to 6 classes, with each class including between over a dozen and almost 200 families. If we're looking at the human genome, there are only 17 food-digesting CAZymes, so most of dietary fibers cannot be digested by human enzymes in upper GI tract and will reach the large intestine for gut microbiomes to digest. That's why gut bacteria typically encode more than hundreds of CAZymes. Some bacteria can have over 6% of its genes encoding CAZymes. If there are 1,000 bacterial species in human gut and each has 100 CAZyme genes, this means over 10,000 CAZymes present in human gut microbiomes, such large dense number in human genome. How do people annotate their microbial genomes and metagenomes for CAZymes? Before 2012, people blasted their query against the CAZy protein sequence database. However, CAZymes are known to be modular and contain multiple domains. In 2012, we built a CAZyme HMM database called dbCAN, which defines a signature domain for each CAZyme family. This allows to identify all CAZyme modules in a given query. After 2017, there are tools developed for short discrete amino acid peptides and the signature of each CAZyme family. One of these tools, ECCB, was published by our lab in 2020. And in 2018, we combined those three ideas and developed a meta server called dbCAN-2, and we found that CAZymes annotated by 2 and 3 tools are more accurate predictions. Here, I'm showing the global map of recent visits to our dbCAN website. In the past 5 years, dbCAN has processed almost 150,000 user-submitted jobs. This shows the top visiting countries for the first 7 months of 2022. This is top cities of US. And these are citations of our dbCAN papers, totaling almost 2,500 times. There are several tools that include our dbCAN as part of them, such as Keybase, Metabolic, and others. Our dbCAN also shows the high rank and citations in database comments compared with other databases. Searching papers citing dbCAN found that it is widely used by agriculture, nutrition, bioenergy, environment, and microbiome research communities. One of the major findings in carbohydrate utilization research is the discovery of polysaccharide utilization loci in late 1990s. The first studied PUL is the starch utilization loci in Bacteroides which contains 3 CAZymes, 2 transporters, 1 regulator, and other like sugar-binding proteins. Since 2005, hundreds of PULs have been described in the literature, while most of them are characterized in gram-negative bacteria. In 2016, PULs were also found in gram-positive bacteria. So, really, PULs are experimentally characterized CAZyme gene clusters in different bacteria to fully degrade carbohydrates. The targeted carbohydrates are major components of plant, algae, and even fungi cell walls. That indicates that CAZymes forming CGCs is general mechanism in bacteria living in any carbohydrate-rich environment. The top ranked genera are from Bacteroides, Proteobacteria, Firmicutes, and Actinobacteria. The top ranked substrates are from plant, but also animal host and bacteria. Obviously, CAZymes forming gene clusters is a widespread phenomenon that microbiomes have evolved to adapt to carbohydrate-rich environments like animal gut, forest floors, soils, and oceans. Determining the substrate of these gene clusters requires experimental characterization and cannot be done in a high-throughput manner. However, predicting these gene clusters can be automated computationally. We have developed CGC Finder, a program to automatically scan any given DNA sequences to identify colocalized TCs, CAZymes, TFs, and STPs. It is integrated in dbCAN tool package. Based on the result, we developed novel substrate prediction approaches. Very recently, I have scanned over 9,000 metagenome assembled genomes, the max, from 4 ecological environments using CGC Finder. We got almost 500,000 CAZymes and 169,000 CGCs. Postdoc Xinpeng developed two methods to predict substrates for these CGCs. The first one is to compare CGCs with 612 POLs in dbCAN POL, which all have known substrates. In this example, the top is the query CGC and the bottom is the subject POL. If we find significant sequence similarity between those two, we will transfer the substrate from the subject to the query. In this example, the substrate is mucin. The second method will be described in the progress on the next page. We have added 3 substrate prediction methods to dbCAN-3 and have published them. We utilized the previous CAZy and dbCAN-HM database and also additionally incorporated the dbCAN-sub-HMM database. This database consists of various CAZymes with EC numbers and corresponding substrate annotations, which were manually created from the existing literature. By employing the dbCAN sub-HM annotation, user obtained the initial substrate annotation for CAZymes that were not present in CGCs. For those CGCs predicted by CGC Finder, in addition to the method mentioned before, we also use another method called the voting method to select the substrate with the most votes that the CGC and the substrate based on the result of dbCAN sub. These are updated on GitHub and also our web server. Here is a list of web links to our dbCAN family's tools. Our dbCAN series currently has substrate prediction methods developed and a database constructed. I want to thank our lab members and Professor Yin on the dbCAN project, and also I'm honored to share our lab research to you all. Thank you.